All right, welcome back, everybody. So today I thought we could talk a little bit about gene set enrichment analysis as far as the whole spatial transcriptomics stuff goes that we've been working on. So we have the genes for a bunch of fields of view, and we are using the 6000 plex. So what we can do is use the human gene sets and look and see, are our cells in our samples being enriched? Which ones are being enriched? Are certain ones being depleted? Are they spatially enriched? So on and so forth. And so the end of this spatial gene set enrichment is going to be kind of working on rebuilding this function specifically for the data that we have inside of this spatialGE package. So this is where it's coming from. We're going to modify it a little bit to work better with our data formats and the data that we have. What we're going to emulate is what I had done for my project, my Cosmics SMI project, which is also here on GitHub, which comes down to three different things. We're going to do the gene set enrichment analysis using the Hallmark Gene Sets from one of these packages, MSigDBr. And then in the end, we're going to permute the positive locations like we had done in the previous video for the spatial clustering to find out if those that are high scoring for our gene sets are closer than we expect if they were just randomly distributed on our samples. So if we come back over to our environment, we can see we were going to import tidyverse and Seurat, and then we're going to need these three new packages that we have not installed yet. And these are GSVA. So here is the Bioconductor page that I'll put a link down in the description. They also have a really nice vignette on how to use this. The data that we're going to use is our input into this is our gene expression for our cells, and then the Hallmark gene sets. Let's see if there is an example. Yeah, not that one. GSVA. Anyway, we'll get to it. The last one that we need in order to do this is going to be this MSigDBr. There is an additional one that I find really helpful when doing big compute tasks like this, which is this mcapply. And what this does is it wraps around mcapply from the parallel package, and it does a progress bar across the bottom. So as your jobs or the list is being kind of gone through, you'll see a progress bar for every single one that is part of the list, which makes it a lot easier to keep track of what is being done and how far it still has to go before you kill it thinking that it's not doing anything. So with those three installed, we can go ahead and load them using library. Now to prep our data, for this, all we need is the gene expression for the cells, and then whether or not those cells are from the same field of view. So we want to only check for gene set enrichment for cells within cells of the same field of view. We don't want to do it across the entire study because there might be more cells of a particular type in a field of view and not in another one, and it can skew the data. So what I did is I imported the Seurat object that we've been working with. So here is the in situ typed data, and then we have the element inside our assay from our SC transformation called scale data, and that's what we're going to be using here. And then I just make it a matrix. After I've done that, I export it. So save RDS, that way if we want to come back later and use our gene expression, we can go ahead and do that. We don't have to import this really large Seurat object again just to get the gene expression out. So that is this one. We just read it in, and then we have our really, really big matrix. It might not even open up, so we'll keep talking. And then the metadata. So within our Seurat object, if we do object at, and then, nope, it's not going to work. Object at, and then metadata. This is what is saved. I saved it out previously. That way we didn't have to, again, read in the entire Seurat object just to get the metadata. So with those two, then we can start moving forward. We have the gene sets. So in order to pull the gene sets that are part of this MSigDBr, we have to specify the species. And since we're working with human samples, we'll specify Homo sapiens. And then out of that, might be easier if I just restart it. But then I will lose everything. From there, then there's a column that's called GSCat, or category for the gene sets, and H means hallmark. So we're going to use the hallmark gene sets. Then what I do is I just get unique combinations of the gene set. So there's 50 gene sets, and then the genes that are associated with that gene set, and just make a really long data table. With that, then we can go ahead and split this table into a list where the names, each element in the list is a different gene set, and then within that element are the genes that are associated with the gene set. So the first element might be adipogenesis, and then within adipogenesis is all the gene sets associated with that. And then I saved that out, and then just read that in. That way, next time if we want to go back and do this, let's go ahead and restart this because it's gonna take a really long time. I'll show, instead of having to read in the entire object again, all we have to do is read in, let's see if it'll let me refresh. Read in those specific elements. Oh, it looked like the R thinking thing might be still spinning up. Our studio session. There we go. So we'll go ahead and do this, and then we'll read this in, and then I can continue talking. So in our metadata, like last time when we were working with the spatial and the spatial clustering, which we can see here in the bottom right-hand corner, we need to specify which cells come from a specific field of view. And if we have several slides like we do here, we also need to concatenate slide and field of view together, because we'll have on slide 1, 2, and 3, there's going to be a field of view 1, for example. We have to make sure that, depending on the study, we have to make sure that no matter what, all of the cells that are being pulled in for this field of view only belong to this field of view, and not field of view 1 on slide 1 and field of view 1 on slide 2. So what I did is I just pulled in slide ID numeric with slide, and then the field of view with FOV. So then we have a new column called unique field of view, FOV. Then from there, what I did is I pulled out all of the unique FOVs into a vector. That way we can kind of sample it later. Not a sample, but iterate over each one, which comes down to this next part. So in PBMCLapply, like I said, we'll get a progress bar. I'm not going to run it here because it does take a really long time. But if we set names for our field of view, then we will get a element that says something like field of view, or slide 1 field of view 1, and the name of that element in our vector is slide 1 field of view 1. And what that makes is the output list has that as the name, instead of it being empty and we have to use a number to grab it. So we can go ahead and read this in, and I can show what I'm talking about. So we have a name here associated with this slide and field of view, instead of it just being like a number in the square brackets like this. Cool. So inside of this, so let's go ahead and I can show what this looks like. FOVs. Probably help if we do this guy. Cool. So we can see the name associated with that vector. So for this, what we're going to do is assign the first one to a variable called FOVID, and we'll run through this lapply function to show you guys what is happening. So now we have FOVID, slide 1, field of view 100. So these first two lines here, what I'm doing is I'm subsetting the metadata that we have to include only cells that are on this slide in field of view. And I'm also subsetting the gene expression to only include the gene expression from cells on this slide in field of view. So we can go ahead and look at that. If we look at FOV meta, we can see that there's only 323 cells now with the 73 variables or columns. And we look at the dimensions of field of view genes. We can see that we have genes by our cells. Cool. Now we can pass in, since we have genes in the rows and cells across the columns, so that's what this means, rows by columns, we can pass this directly into the GSVA function. We don't need to transform it like we would if we had cells in the rows and gene expression, well, genes in the columns, gene names. By passing it into GSVA, we also need to pass in our gene set list, which is going to be the Hallmark Gene Sets and then the genes that are associated with the gene sets. So let's go ahead and run that. And by doing this, it's going to calculate the gene set enrichment score for all cells for all gene sets. So it's a very fast function. You can see that it's done already for this field of view. That's all the longer it took to calculate. Let's go ahead and look. Cells GS. A big matrix of gene sets by the cells, which is cool. So we'll save that one. Now what we want to do is we want to look at the different cell types that are on this field of view. So that's going to be this line here. And what we're going to do is we're going to collapse it down. So all of cell type A that are on this field of view, what is the gene set score that it got for every single one of the gene sets? So first we need to identify the phenotypes. So A, B, D, H, M. And then we're going to set the names like we did above. That way when we run our lapply, the names will be there. So now if we look at Fen, we can see that the names are above there. So our list will have the elements called B, D, A, H, for example. Within here, we're going to iterate over those, and then we're going to pull out of our cell GS the cells that are of group B, for example. After doing that, we're going to do the row means. That way we have one score associated with B for every single gene set. So what we can do is run this, and then we'll do the row names too. That way we can see what those look like. And then check this out. So now we have a table of cell types, phenotypes, and then all the gene sets. So this here, -0.1, is the average of all cell type B for adipogenesis. And then later on we can go and compare all of cell type B and all of the FOVs from this treatment to all of cell type B and all of the FOVs from this treatment. Are their scores different for adipogenesis? And there's better ways to do that with repeated measures, but this is one way. And then I'm returning the cell type sets by cell type scores, and then by cell scores, and then by cell type scores. And that's in that really big list that we had seen here, where each element, now if we look inside, has two parts. We have a table that's cells by gene sets, so rows, columns, and then we have phenotype sets where we have all the phenotypes that are on this field of view and the average of each gene set. So there's 50 gene sets. B gets 50 scores. Cool. Now what we can do is go ahead and plot this. So what does the gene set score look like for cells on a field of view if we plot the locations, so the X and the Y location, and then the color based on a gene set score. What I'm doing in this first part is we're going to select a field of view, and in this case we'll just do the first one, and then we're going to pull out the cell sets. So this is going to be all of the cells in that field of view, the scores that they have for all the gene sets. My dog is scratching her ear, making weird sounds. This is a matrix, so we can go ahead and look at 1 through 5 and 1 through 5. So we have gene sets by cells. What I'm doing next is I'm going to make it a data frame table. I'm going to make sure to check names equals false, because sometimes if you have weird cell names, it might try to change them, so you can't have a space. If you do check names and you don't put anything in here, if you have a space, it's going to put a period there, and then you're not going to be able to merge it later on. Next, I'm making sure that our gene sets are actually inside of our table. Right now they're rows, but we want them to be inside of our table. And then I'm gathering by removing the gene sets names. Let's go ahead and look at this. Let's see if it works. It's a little not pretty. So we'll do tibble, and then we'll feed it into head. There we go. So we have a column for our gene sets and then all of our cells. What I'm going to do next is rotate from cell and score all the way over into long format. So we're going to move from wide to long, and this will allow us later on to filter based on the gene set that we want to plot. Makes it a lot easier. So that's what this gather is doing. We're going to ignore our Hallmark gene set, and then we're going to gather where the column names are going to be our cell ID, and then the numbers are going to be in a column called score. So if we look at this, I got to run it because I restarted R. There we go. And go up 2, and there we go. So now we have our score, our gene set, the cell, and the score, and we can see that this is all the same cell, and we're calculating the different scores for the gene sets. Next, what I'm doing is I'm making sure that the metadata that we have only includes the cells that are from our Hallmark scores. So by doing this, we're just going to subset it down FOV meta to our 323 genes cells. That way, in this step, we can merge them together. That way we have our metadata along with the scores. So by opening this guy up, we can see that we have our metadata in the first 5 columns, and then we have our gene set and score. So this is where I was talking about. Now we can subset down to include only a particular gene set that we want to look at. So let's go ahead and do a different one because this is the example. Apoptosis is a good example. Let's go back over here. We can look at all of the unique gene sets by going this way as well, and we'll do apoptosis. I don't expect to be too much spectacular, but we'll see. So I'm going to filter to include only the apoptosis scores for this sample. We're going to do a point plot where we're going to look at, might be way over there, the X and the Y location. Yeah, here we go. We're going to use the X and the Y location to put our cells in our sample. And then we're going to color it based on our score. So this is the score for our cells. And then I'm scaling to lowest score being blue, highest score being red, just to make it look a little bit better than what default R would be. And then making sure the X and the Y coordinates have the same scale, but more or less. So here is the output. I don't see anything that really stands out. Looks like the really low scores are kind of scattered everywhere. Some of the high scores are a little bit clustered, but it's not a great scale to see. But we'll save the spatial part for next time. Thanks, guys.